So, as we have just discussed in last lecture, that what are the Bergey's manual and how the Bergey's manual defined the diversity from phonetic to basically the phylogenetic basis. So, all characters on the basis of the phenotype, on the basis of the phylogenetic analysis, on the genome, on the genetic variations, on the mutations, we categorize into different categories, and Bergey's manual endorsed them into 5 different volumes. Volume 1, 2, 3, 4, 5. And we just discussed in my previous lectures that volume 1 carries certain kind of a bacteria having certain characteristic features, then volume 2, volume 3, volume 4, and volume 5. So this is a comprehensive Bergey's manual, provide the all organisms, microorganisms, parasites, pathogenic food producers, beneficials, industrial applications, medical applications, all being categorized into different categories, different groups, and that can be easily approached. The most important thing is that through this Bergey's manual, you can approach the microorganism to learn and to understand the characters and to make use of it. But besides that, what are the challenges of this diversity? How this diversity will cause a facing a problem basically for us. So one of the most important greatest challenge before the scientists, before the microbiologists or taxonomists in bacterial taxonomy is that the sheer scope of microbial diversity that remain undiscovered. There are many bacteria, there are many microorganisms which are not discovered, which are not known, but they exist. And that's how we talked in earlier, I think one of the lectures we talked about the metagenomics. So metagenomics help us to understand those bacteria which are not discovered, because they cannot be cultured. Those microorganisms which can be streak out and can be cultured, well, they can be understood, they can be understood. But I think those microorganisms which cannot be cultured, we cannot discover them, we cannot look into, we cannot visualize them. So that would be very difficult. So this is one of the major challenges. So almost 2,500 in class of these microorganisms are species, approved name, bacterial names like retained in the approval list from the tens of the thousands in the historical literatures. And less than 5% known species estimates percentage of bacterial species currently identified in the nature. 1923, the first manual, Bergey's manual, uh, David Bergey, who first, jot down the manual, published the first bacterial classification manual to define all these species. But there are many species. There are many bacterial species which are not even known, not even aware about it. Right. So besides, it's not 2,500, but there's a millions of different species of it. So if you look into the approved list versus validation are valid publications, then we can compare these two. So we call them as the approval list or approved list versus the validation or valid publication. The bacterial nomenclature system distinguish between the approved name and validity published name. The approved list contain almost 2,500 name. Now, which retain from the historical literature because they are published in the Bergey's manual as the first manual, then second, and on. And new bacterial names must be valid, published in the International Journal of Systematic Bacteriology, are announced in the validation list to gain standing in the knowledge. What does it mean? It means the name which is given to a particular organisms and endorsed or jotted down or written in the Bergey's manual should also be internationally known, right? So that has to be published in International Journal of Systematic Bacteriology. Now you have to validate that. What is the validate? Validate means like as the characteristic feature of the classification is the predictability and stability. So the whole classification is based on predictability and stability. Now, if you remove these two characters or two reasons rather, predictability and stability, then classification has no meaning, right? So classification has only meaning when you keep intact the stability and predictability. Because if the classification, mode of classification, whether it has been written in the Bergey's manual or whether it has been written in some other manual, I mean, the character should be stable throughout this time scale. Now, if these characters are not stable, then probably it's very difficult for you to identify or to predict basically these things. Now, so we define them as approved lists, historical names approved for retention in the bacterial nomenclatures. These 2,500 names were salvaged from tens of thousands in the literature because they could be clearly defined and linked to specific bacterial what has been defined in the Bergey's map. Now, validation list means the new name that are validly published and must be considered in the bacterial nomenclature. So there's also criteria for defining the nomenclature. How do you define the bacterial nomenclature? How do you name the bacteria? For example, Escherichia coli, Klebsiella pneumoniae, Pseudomonas aeruginosa, Pseudomonas putida, A. baumannii. So what is the basis and what is the criteria and the rule of nomenclature? How to name it? Because if you are not going to name these bacteria, then it would be difficult internationally and globally to pick up the bacteria from even define anywhere. So nomenclature should be uniform, should have a uniform pattern, should have a uniform rules and regulations, and should give a single name of a single species with the common characters. However, the scientific community may choose not to adopt name if the evidence is insufficient. It means though you try to publish it, but I think scientists will not consider it, will not validate it until it has a common characters, exactly the same characters were identified to that particular individuals whose name already been defined. If there's a variation, name may be changed. Scientific judgment in taxonomy. How the scientists judge and how do scientists, explain the taxonomy, especially bacterial taxonomy, which is ultimately relies on the scientific judgment. So if a scientist doesn't validate, doesn't accept the fact, then probably that name or that species will not be considered in that published or approved list. So bacterial taxonomy ultimately relies on the scientist's judgment, scientist validation, and community consensus, right? Rather than the rigid rules. So we should not follow the rigid rule, but we should have like some consensus to decide the name, to decide the category, to decide the place where it has to be kept in the hierarchy of the classification. The acceptance of a new bacterial name depends on the strength of a scientific evidence supporting the proposed taxa. In particular taxa, what are the strong evidences, right, which tally, and these evidences which tally to the concerned taxa should be included before the name is given. For example, Helicobacter pylori success, for example, immediately accepted as a placement of a Campylobacter pylori due to strong molecular evidences between the variation from the Campylobacter pylori to Helicobacter pylori. Now, it has been taken. It has been taken easily. So this is called, we call it the success story of the Helicobacter pylori, which is accepted by the scientists very successfully. Now, Tetlochia mcdaddy is rejected. The name is rejected because it was not found, so not generally accepted because a replacement of the Legionella mcdaddy. So the name was replaced by Legionella rather than Tetlochia because of the consensus of the community and because of the some strong features and the consensus and the judgment of scientists with the despite of valid publications, they have changed the name. So this is how the over a period of time, or with some meetings and consensus, the name can be changed based on certain rigid characters, but not even the published list which is already there. Now, the microbial evolution, the RNA world hypothesis, is very important here. What is the RNA world hypothesis? The RNA world hypothesis suggests that the pre-cellular life was dominated by RNA molecules that could both store genetic informations and catalyze chemical reactions. So this is how it works. Now, the RNA bird hypothesis suggests that the pre-cellular life was dominated by RNA molecule. It means the defined RNA molecule in the genome. We are talking about the bacterial genome, which has a DNA as a genome, but they have RNA molecule also that could both store genetic informations and catalyze the chemical reaction. So it means there's certain RNA gene which can also involve in the metabolic processes. RNA catalysis, RNA molecule like ribozyme, which can catalyze the RNA molecule, which is catalyze the reaction essential for metabolism, right? We call ribozymes. Ribozymes are the only enzymes which can have RNA as a component. It is non-proteinaceous enzymes. They have all turnover number, they have a catalytic activity, they have kinetics, but they are not proteins. The exceptional enzymes which are not proteins. Otherwise, all enzymes are protein. All microbial enzymes are protein, except RNA enzyme. Now, RNA replication. So RNA can store and transmit genetic information through self-replicating. So this is again the role of RNA. RNA, for example, many virus RNA viruses, they all replicate. For example, mRNA virus are mRNA plus, mRNA minus. So those minus strand mRNA viruses first convert into the plus strand mRNA viruses. Then from plus strand to the transcribe into the another RNA to for the expression of a proteins, and again they form the negative strand. So this kind of RNA world, the strength of RNA is very important for the survival of the microorganisms and for the metabolic processes and the replication cycle of the microorganisms. DNA evolution, DNA eventually replaced RNA as the primary genetic storage molecule. So earlier it was RNA, but now later on it was changed from RNA to DNA, which is more advanced form of the nucleic acid. On the three domains of life, as we already discussed in earlier lectures, that is bacteria, archaea, and eukarya. But how we can look into in terms of like a classification, determination and characteristics of a classification. So all living organisms can be classified into three fundamental domains, as we already talked a lot on that. These three are bacteria, archaea, and eukarya. Bacteria are prokaryotic organisms with a peptidoglycan cell walls and diverse metabolism and circular chromosomes, and mostly numerous and metabolically diverse biofilm formations. Whereas archaea are prokaryotic organisms which are not exact prokaryote, but I think they are under the prokaryotes with the variation from the bacteria, which are also prokaryote. The different cell wall compositions. Often they are extremophile, as we already discussed in detail in earlier lectures, with the molecular machinery more similar to the eukaryote. So this is the border at the border between the prokaryotes, that is the bacteria, eubacteria, and the eukaryote. And eukarya organisms are the multicellular, from unicellular to multicellular membrane-bound organelle producing organisms. Now, polyphasic approaches to microbial classification, although I have already discussed about the polyphasic approach in my probably the earlier lecture, because we are all talking on the series of the microbial diversity and the characteristic features and the classification and the determination of classifications and the mode of classifications. So the modern microbial classification employs a polyphasic approach. Polyphasic approach means you can have a multiple approaches like genetic analysis, phenotypic analysis, phylogenetic analysis. In genetic analysis, DNA and RNA sequences provides the molecular foundation, understanding of evolutionary relationship in the species boundaries, looking how these genomes are modified, how these genes in the genomes and the chromosomes are changed and evolved and mutations taking place through horizontal gene transfer, through random mutation, through spontaneous mutations, right? So over a period of time on time scale, genetic change is taking place. Now these genetic changes were analyzed and help us to categorize these organisms in different categories. Now, phenotypic analysis, these are that overexpression of the phenotype, that is the phenotype of organisms, the expression characters which can visualize by naked eyes or to some extent in the microscope, and the morphological and physiological changes, biochemical characteristic features in the form of some byproducts, right? And which help in identifying the criteria for conjugate. Now, in case of phylogenetic analysis, basically phylogenetic analysis is based on both phenotypic and genotypic characters, but on the evolutionary scale, on the time scale, these characters when keep changing over a period of time, right, then it make the organisms indifferent. For example, if you talk about the E. coli, now if we more minutely describe E. coli within the different within the E. coli space, different strains, then we found that there's thousands of different strains were found. Now, Probably we'll talk in the next lectures, maybe tomorrow, on that. So this evolutionary changes in the mutations taking place make them at different types and different strains within the same species, which is at the very narrow hierarchy level, that is the species. And this evolutionary relation revealed to different phylogenetic analysis. So you have to characterize different phylogenetic analysis. These organisms are being kept at different branches of the tree as it evolved, different hierarchical organizations in the taxonomic group. So we call them as a phylogenetic analysis or phylogenetic characteristics. Now, if you look into the taxonomic hierarchy, there are seven broad classification, seven broad space where these are defined. So domains is on the top. So first we have classified the domain, which is the broad classification. And then from domain, there are three major groups of lives. Then come to the phyla, which is a large group. So all the characters of the domain, single domain, must be present in that phyla. Right? Phyla is a kind of a subset of a domain. And phyla, they are large groups with the fundamental similarities to different domains. Now, similarly, the class which come under the phyla, so you have a multiple classes in the phyla. So a lot of features which are defined in the phyla are found in the classes with a little variations. So we call them, this is one class, second class, and third class, and so on and so forth. Then class, then come to the order. So many orders which are under the classes. Then families, which is related genera. So it means different species of the same kind form one genera, different genera, right, form different families, they form different orders, and different orders, they form different classes, and different classes, they form different phyla, and different phyla, they form different groups. So this is the whole hierarchy of the classification of these organisms on the basis of different characters, both molecular characters and phenotypic characters. Now, classical versus the modern taxonomical approaches. The evolution of the bacterial taxonomy reflects the transition from classical morphological and physiological approaches to modern molecular methods. The classical taxonomy relies on observable characteristic features or characteristic methods. These classical taxonomy relies on the observable characteristic like cell wall, cell shape, staining properties, that's we define gram negative, gram positive, and various other metabolic processing physiological changes. Now, if you compare the modern method to the classical method, and we find the variation in these two methods to categorize them into different, hierarchies group. Morphological characteristic features, physiological features, metabolic features, then ecological preferences where the organisms prefer to live, staining reactions, and so on. Now, many of them, they are common. For example, if you talk about the metabolic capabilities and you talk about the physiological properties, both are hand in hand, both are overlapping because metabolism defines physiology, and physiology is basically on the basis of different physiology, the metabolic output is coming. Now, if you look into the modern methods are DNA, RNA sequencing, the best method, phylogenetic analysis, comparative genomics, molecular markers, and protein analysis. Now, what are the protein analysis and what are the RNA DNA sequencing? We'll discuss later on. Now, the challenges in a horizontal gene transfer. The horizontal gene transfer is also playing a very, very important role in changing the species, in changing the different classes, keeping them into different categories. For example, plasmid, which is extra chromosomal genetic material, carrying different gene, different transposon, different IS elements, which can help on changing the mutations, variations at a different time scale. So plasmid bond traits can cause taxonomic errors, uh, if not because it carries different genes, if not, then carefully distinguished from the chromosomal characteristic features. Conflicting signals. Different genes may suggest different evolutionary relationship. Right. So you can have a different signal sometime within the same, intact individual's genome due to independent transfer events. Core versus accessory means like distinguishing between core genome, that is the main genome, vertical inheritance, where the genome divide through mitosis and meiosis, and then can differentiate into two different cells. And accessory genome means like horizontal gene. For example, the gene transfer through plasmid, through transposon, right, through IS elements and through other things. Now, the future of bacterial taxonomy. The future of vector taxonomy lies in the integrating the increasingly sophisticated molecular techniques with the traditional approaches. Whole genome sequencing. So the whole genome sequencing is the breakthrough. Metagenomics is one of the most latest and advanced techniques where we can comprehensively identify even those microorganisms which are non-culturable, even which cannot culture in the lab. You just take a sample, a clinical sample, water sample, soil sample, or any environmental sample, and get the DNA out. So there are many kits are available now in different companies, and you can have some methods. We can directly from that sample you can directly isolate the DNA. You need not to isolate the first microorganisms by culture, as in case of Enterobacteriaceae or some culturable bacteria, which are very less in terms of like a percentage. And you can get to whole genome sequencing through metagenomes. Whole microbiome can be sequenced. So in a microbiome, you can have a multiple different type of bacteria which are not even found earlier, non-culturable, and so on and so forth. So these are the most latest technology. Complete genome analysis. Then we have a big data. Now, what is the big data means? Big data means the genome which generate the huge amount of data, and this huge amount of data which has been generated can be analyzed, can be capped into different classes, different categories, different categories, and then make use of this big data by through analysis. There are many softwares, computational based softwares are available. You can use these softwares and you can analyze the data. Right. By analyzing the data, you can again further identify several. No, these big data can help to integrate into the artificial intelligence, like latest algorithm, ANN, artificial neural network, then SVM, then many other algorithms are available, Monte Carlo, which can help you out to learn about the AI integration. So how machine learning classification, which is totally dependent on the big data, are dependent on the data. How refined your data is, how big the data is, how uh, fair the data is, right? Purified the data is, I would say, without any contaminations, then you can have better AI model. So you can generate the AI model, prediction model. Uh, you can predict, many things. I'm not specifically talking about some specific thing, but just have an example. COVID-19. So COVID-19 is basically pandemic came 19 to 20, 21, 22 till in this era. Now does COVID-19 pandemic come again in next 5 years or 10 years? Can be predicted through AI. So for that, you need to have the big data. From where the big data comes? From the whole genome. So you can have a whole genome of the viruses all around the globally, get the data, identify them, then develop a model. Bigger is the data, more is the data, better the PI prediction model. So very simple. Now, what is the method to generate this data? As I've told, metagenomics. So environmental diversity, for example, if you take the sample from the environment, so you can have a multiple different types of viruses, taking the same example of COVID-19, SARS-CoV-2 virus, and you can have a varieties of the data. And this data will be used for the AI integration. Then for the understanding the functional interaction of these output of the genome, that is the protein output or the gene product output, what is the system biology. So system biology is integrated functional analysis. In system biology, you learn the outcome of the protein, outcome of the gene. Now the gene outcome is basically the product we call the protein, and that protein, what is the function of protein? Although you can also use the proteomics analysis, right? Like genomics, you also have a proteomics analysis. The proteomics analysis can give you the differential expression of this protein in the microbial flora. Now, why I am talking about this, because this is the ultimate, this is the future of the microbiology. This is the future of the bacterial taxonomy. So the taxonomy, which was started in like early 20th century or late 19th century, people, philosopher and scientists, they jot down these microorganisms on the basis of certain characters. Now we are in the era of the genomics or the metagenomics or AI, where we can use these characters, develop a big data, and make use of this big data to generate AI model. And this AI model will help us to even further categorize them into more minutely these microorganisms in different classes, categories, and hierarchy. So we can, in other words, we can say that we can make more better mode of classification, more better way of identification of these microorganisms. For example, if I say you pick up the swab of the oral cavity like this. Now, this swab of oral cavity carries certain different microorganisms. You identified them through conventional method, but you will not find all. But if you go through metagenomic analysis, this metagenomic analysis will help you to understand every microorganism which are even non-culturable microorganisms, which through normal swab cannot picked. Now, once you got these non-culturable microorganisms and we got diversity, and you understand them what these microorganisms are, you can pick up some new kind of a bacterial species, with the new characteristic features, and that new characteristic features can be utilized for the prediction, future predictions. So this is what the future of bacterial taxonomy. Now, having that characteristic features, identification of the bacteria, microorganisms, viruses, fungus, parasites, and all in different classes, make use of their medical use, application, industrial use, agricultural use, right? And in order to refine this uses, application of these microorganisms, the AI could have very important role for prediction, both for disease predictions, their benefits predictions, their characteristic features, to know it, how this bacteria can be used for the good crop production, good vine production, good food productions, and so on and so forth. So this is how the importance of the bacterial taxonomy, and most of what is the future of bacterial taxonomy. Now this is all we all done. Thank you very much. And now we would have another lectures on some more advanced form of this. Now thanks a lot.